Welcome to another deep dive, everybody. This time we're venturing into the fascinating world of big cat brains. We're going to unpack some research that's giving us a look into how these majestic creatures age, specifically their brains, and the potential for Alzheimer's-like changes.
Yeah, exactly. So this study was published in the Journal of Comparative Neurology in 2024, and it looked at brain tissue from 9 individual animals across 5 different felid species.
Okay, cool. So who are these big cats we're dealing with?
Well, the lineup includes cheetahs, clouded leopards, African lions, servals, and Siberian tigers.
Oh, wow, that's a pretty impressive group. And they ranged in age from 15 to 24 years old. Sounds like we're focusing on the golden years here.
Definitely. And it's worth noting the evolutionary relationships between these species. The lion and Siberian tiger are the most closely related, which kind of makes you wonder if closer relatives might show more similar brain aging patterns.
Interesting. Now, let's get to the heart of the matter. What were the researchers actually looking for in these felid brains?
So they were focusing on 2 main culprits that are often associated with Alzheimer's disease in humans: amyloid-beta, or A, and tau protein.
Right, I've heard of those. They can clump together in the brain, right?
Exactly. And those clumps can disrupt normal brain function.
And to be more specific, the study targeted 3 brain regions: the gyrus sylvius and the CA1 and CA3 subfields of the hippocampus areas that are particularly vulnerable in human Alzheimer's.
They didn't just look at amyloid-beta generally. They actually distinguished between 2 specific forms, A40 and A42. And they also examined A in 2 different locations, in plaques, which are those clumps we talked about, and in blood vessels, that's called vascular age. All of these distinctions can have different effects on the brain.
Okay, so there's a lot more to A than meets the eye. All right, drum roll, please. What did the researchers find when it came to A in these big cats?
Well, the surprising thing was that every single felid in the study, all 9 of them, had some degree of A accumulation in their brains.
Really? Every single one? That's unexpected.
It is, but there's a really important caveat. All of the A plaques were diffuse plaques, not the dense core type that's strongly associated with Alzheimer's disease in humans. This diffuse pattern is actually pretty similar to what we see in healthy aging human brains.
Oh, okay. That makes me feel a little better. What about those different forms of F, though? Did they find anything interesting there?
They did. While both Aβ40 and Aβ42 were present, they found that Aβ42 was more commonly found in plaques, while Aβ40 seemed to be more prevalent in blood vessels.
Huh, that's interesting. Does that align with what we know about F in humans?
Well, that's a great question. In humans, Aβ42 is considered stickier and is more likely to form those harmful clumps. So it really makes you wonder what this might mean for how AF affects felid brains.
Yeah, a lot to think about there. Now let's move on to tau protein. What do they find out about tau in these big cats?
Well, remember that tangles of tau protein, called neurofibrillary tangles, or NFTs, are a hallmark of Alzheimer's in humans. But in these felids, tau pathology was much less common than LOC. In fact, only the cheetahs and clouded leopards showed any evidence of tau in the brain regions they studied.
So it's less common than AP, but still present in some of the animals. What did that tau pathology look like? Were they full-blown NFTs?
Not exactly. They mainly found what are called pretangles. That's like an early stage of tau accumulation, not the full-blown tangles we see in Alzheimer's.
Interesting. Anything else about the tau findings that stood out?
Yeah, actually, a couple of things. First, they found more tau in the hippocampus than in the cortex, which is different from the usual pattern in humans and domestic cats. So this brings up the question of whether tau pathology might progress differently in wild felids. And second, the cheetah with the highest density of pretangles was the youngest of the group. Yeah, which suggests that age might not be the only thing influencing tau accumulation. There might be something about the cheetah's biology, their genetics or even their lifestyle, that makes them more susceptible to developing tau pathology, even at a younger age.
That's fascinating. So we've got these distinct patterns of OM and tau, but the researchers didn't stop there. They also took a look at another important player in the brain, microglia. Can you tell us about those?
Sure. Microglia are basically the brain's immune cells. They're constantly scanning for signs of trouble, like the accumulation of O or tau. When they detect a problem, they can change their shape and behavior, going into what we call an activated state.
Okay. So how did the researchers assess this microglial activation in the felids?
They looked at the shape of the microglia under a microscope. They categorized them as ramified, intermediate, or amoeboid, with each shape indicating a different level of activation.
Gotcha. So what did they find out about microglia in these felids?
This is where it gets really interesting. They found a strong link between microglia in an intermediate state of activation and two things: A, accumulation in blood vessels, and the amount of tau pretangles in the hippocampus.
Okay, so there's a connection between activated microglia, vascular A, and tau pretangles. Is that a good thing or a bad thing?
That's the million-dollar question. It's a tough one. On one hand, microglia can actually help clear out those harmful proteins like A and tau. So their activation could be a sign that they're trying to protect the brain. But on the other hand, microglia can also contribute to inflammation in the brain. And chronic inflammation can actually be detrimental, potentially making things worse in the long run.
Oh, I see. So it's kind of a double-edged sword. The microglia could be helping, or they could be making things worse. It's tough to say at this point, huh?
Yeah, absolutely. We definitely need more research to figure out what role microglia are playing in felid brain aging. So we've uncovered some pretty interesting details about amyloid-beta, tau, and microglia in the brains of these big cats. And it seems like the patterns we're seeing are quite different from what we typically observe in humans with Alzheimer's.
Yeah, it's definitely not a clear-cut parallel. Those diffuse plaques, the less common tau tangles, even the different distribution of tau in the brain. It's all suggesting that felid brain aging has some unique aspects. So where do these findings fit in the bigger picture of brain aging, especially compared to humans and other species?
Well, it's a bit like piecing together a puzzle, and this study is giving us some crucial pieces, but we can't just assume that what we know about Alzheimer's in humans will directly translate to other species. This research really highlights the importance of studying brain aging across a wide variety of animals.
Right, because each species has its own unique evolutionary history, its own set of genes and environmental pressures that have shaped how its brain ages. Makes you wonder, could certain felids be more or less susceptible to developing cognitive decline as they age compared to humans or even domestic cats?
That's the burning question, isn't it? And while this study can't give us a definitive answer, remember that only the cheetahs and clouded leopards showed tau pathology. That might suggest that there's a species-specific vulnerability to tau accumulation. It's almost like each species has its own fingerprint when it comes to brain aging, reflecting this complex interplay of genetics, environment, and lifestyle.
Speaking of complexity, we've got these distinct patterns of ASAR and tau, and then we throw microglia into the mix. It's getting pretty intricate.
It really is, and we're just starting to unravel this intricate dance between all these different factors. This study is really just a first glimpse into brain aging in felids. It's raising more questions than it answers.
Which is a good thing, right?
Exactly. It's fueling further research and pushing us to think differently about how brain aging works across the entire animal kingdom. And what's really exciting is the potential for this research to inform our understanding of Alzheimer's disease in humans.
Yeah, even though the patterns of pathology are different, there might still be valuable lessons to learn from these felids.
Precisely. By comparing and contrasting how brain aging unfolds in different species, we can start to tease apart those fundamental mechanisms that contribute to cognitive decline. It's like having multiple natural experiments running in parallel.
So by studying felids, we might gain insights into why certain brain regions are more vulnerable to aging, or why some individuals seem more resilient than others. And those insights could then lead to new strategies for preventing or treating Alzheimer's in humans. It's amazing to think that these big cats could hold clues to unlocking some of the mysteries of human brain aging.
Well, that's the beauty of science. You never know where the next breakthrough will come from. And this study is a perfect example of how studying the natural world can lead to unexpected and potentially life-changing discoveries.
So we've covered a lot of ground, from plaques to tau tangles to microglial mysteries. It's been a truly fascinating deep dive. What would you say struck you as the most surprising or intriguing aspect of this study?
That's a tough one. There were so many fascinating details, but I think what really stood out to me was that link between activated microglia, vascular A, and the tau pretangles.
Yeah, trying to disentangle cause and effect there and figure out whether it's ultimately beneficial or harmful is like navigating a tangled web.
Exactly. It's a puzzle within a puzzle, and that's what makes it so compelling. It highlights just how incredibly complex the brain is and how much we still have to learn.
Couldn't agree more. Even with all the strides we've made in understanding the brain, we're really just scratching the surface.
And that's what keeps me going as a scientist, that constant pursuit of knowledge and the thrill of uncovering new mysteries.
It seems like brain aging is a really complex process, and there's no simple answer. But even with all the complexity, this study gives us a good starting point for trying to understand how aging might affect these animals, and it underscores how important it is to study brain aging in a wide range of species.
Absolutely. We can't just assume that what we see in humans will apply to other animals. Each species is unique.
Right, each with its own evolutionary history and its own set of genes and environmental pressures that have shaped how its brain ages. So we have this really interesting glimpse into felid brain aging. But what's next? What are the next steps in this research?
Well, this is really just the tip of the iceberg. There's still so much more to learn. For one thing, we need larger sample sizes, more individuals from each species to really get a handle on the variation that exists.
Yeah, more data is always good. And ideally, it would be amazing to be able to follow these animals over time, tracking their cognitive abilities and seeing how those changes relate to the buildup of As and tau in their brains.
Exactly. Longitudinal studies like that would be so valuable. They could help us figure out when these changes start to happen and how they connect to actual cognitive decline.
It would be like having a window into their cognitive journey.
Yeah.
And it would also be really interesting to compare brain aging in captive felids to their wild counterparts.
Oh, definitely. That's another big question. Is there something about living in captivity that changes how their brains age? Do they age faster or differently?
So many questions to explore. It feels like with every answer we get, we uncover even more questions.
But that's what makes this field so exciting. It's a huge, unexplored territory, and we're just starting to map it out. This is just the beginning of a fascinating journey.
Well said. Maybe these incredible creatures will help us unlock the secrets to healthy brain aging for ourselves and for future generations.
I like to think so. We've covered so much ground in this deep dive. It's been an amazing journey into the world of big cat brains.
It really has. We've learned about plaques and tau tangles and microglial mysteries. And hopefully everyone listening has come away with a new appreciation for how complex and amazing the brain is and how much we learn by studying it in all its diverse forms.
If you're interested in learning more, we've included links to the original research and other resources in the show notes. And remember, from Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.